Hello guys, so let's take a look at the 2025 WAEC biology specimen that has been released. Now, specimen A is bean weevil. Bean weevil. Now, the things you need to know about this specimen is number one, the scientific name. What's the scientific name of bean weevil? Now, the scientific name is Acanthocylidis obtectus. Now, when you write the scientific name this way, please underline separately. The general name must start with a capital letter and the species name a small letter. That's very important. Other things you need to note is the kingdom. It is under kingdom Animalia, under phylum Arthropoda, the largest phylum, and class Insecta. Okay. Now, aside that, there are other informations you need to know. There are other informations you need to know. And what is that? This particular specimen exhibits complete metamorphosis. Complete metamorphosis. So, in case you are asked, it exhibits complete metamorphosis. That means it goes from the egg to larva to pupa and to the imago, or you can say adult. Okay, so that's that. This exhibits complete metamorphosis. Secondly, you also need to know that they exhibit biting and chewing mechanism. So in case you are asked, what kind of feeding mechanism does specimen A exhibit? It exhibits biting and chewing mechanism. Okay, so that's that. Now, another thing is that they have exoskeleton. They have hard exoskeleton. Hard exoskeleton. Hard exoskeleton. So, now, having looked at these, let's look at other questions that can emanate. Now, you can be asked to draw the specimen. You know how to draw. You have to draw the specimen. If you have been given a size, you have to draw within that size and put your title and your magnification. Very important. Now, let's look at other things we need to know about this specimen. So they may ask you, what's the economic importance? Economic importance of specimen A. Economic importance of specimen A. Now, economic importance can be both negative and positive. Okay. As a matter of fact, in this case, it has to be the negative economic importance. So the first one is that it destroys crop, or you can say destroy the bean seed. The bean seed destroys the bean seed. It reduces crop yield, reduces the crop yield. It um, causes escalation of prices or price of bean as a result of infestation. As a result of infestation, and so on. Okay, so these are the economic importance of specimen A, in case you are asked. Now you can be asked to list the observable features. So when you look at your specimen, what and what do you observe? So you can be asked to list observable features of the bean weevil. Okay, now if we are looking at observable features, the first one that should come to our mind is that since it's an insect, it is divided into three parts. Okay, so divided into three parts, which is the head, the thorax, and abdomen. The head, thorax, and abdomen. That's the first observable feature. Now, secondly, they have hard exoskeleton. Hard exoskeleton. They are relatively small inside. Relatively small in size. Um, they possess compound eyes. Possess compound eyes. They are oval in shape. They are oval in shape. They possess or they have three pairs of jointed appendages or legs. So these are few observable features of these specimen. Okay, so these are the questions that are likely to come out under specimen A. All right, specimen B is bean seed with holes. Now, the first thing you should know about specimen B is the botanical name. So the botanical name of specimen B is Phaseolus vulgaris. That's the botanical name of beans. And the kingdom is kingdom plantae. So what are the questions that can emanate from this particular specimen? Number one, they can ask you to list the observable features. Observable features. So when you look at the specimen, what can you observe? Firstly, there are holes. Hmm? Holes on specimen. Okay, or holes on the body of specimen. Secondly, you can observe that there are powdery residues. Powdery residue on specimen. You can also observe that there is discoloration. Discoloration of specimen. Then probably they have loose texture. Loose texture. So these are the major observable features you can find on this specimen when it's provided for you. Now, if you observe that there are weevils inside the holes of your specimen, you can as well put that down because it's possible to find weevil inside the holes of specimen B. Okay, so that's that. Now, another question that can come out from this specimen is they can ask you what kind of germination is exhibited by specimen B. So specimen B exhibits they exhibit epigynous germination. Okay, they are dicot plants. So plants with two germinates with two seed leaves carrying the cotyledon above the the soil or above the ground. Okay, so that's that for specimen B. Now, they can also ask you what is the relationship between specimen B and specimen A. Remember, specimen A is being weevil. Okay, so they can ask you what's the relationship between specimen A and specimen B. So it is very simple. Specimen A parasitize on specimen B. Okay, so specimen A parasitize on specimen B. The moment the adult lays the egg develops into larva. The larva start boring the holes in the beans, okay, and start feeding on the beans, and after which it turns to an adult, and that is how the cycle continues. So that's the relationship between specimen A and specimen B. So specimen A causes this condition. So the specimen A is what causes the holes in specimen B. So that's that. And they can also ask you the preventive measures. What can you do to prevent the holes on specimen B? So if you're looking at the preventive measures, you can say store in a cool, dry place. Do not store in porous bag or open bag. So don't store in bags that has pores. That's what that statement means. Now, another preventive measure is you can use natural repellent. I wouldn't advise insecticide because it is something you want to eat. So use natural repellent like the neem leaf, the neem or bay leaf. So the neem or bay leaf are natural repellent. So you can use that for preventing infestation. Okay. Now, other things is that you can check regularly for infected seed and separates. So you can check infected seeds regularly and try to separate them to avoid further infestation. So that is what these are the things you have to have in mind, or these are the questions that you are likely to see under specimen B. All right, so specimen C is water leaf plants. So the first thing you need to know is the botanical name, which is Talinum triangulare. So that is the botanical name of water leaf plants. So what are the questions that can emanate from this particular specimen? Firstly, okay, you should just know that it is under kingdom, since it's a plant, kingdom Plantae. So the questions that can emanate from this specimen, they can ask you to pick out a leaf of the specimen and draw. Okay, so they can ask you to pick out a leaf of the specimen and draw. So you can pick out the leaf of the specimen and draw, include the venation. Now, you might be asked, what kind of venation does specimen C exhibit? Now, looking at this venation, it falls under the net-like kind of venation, but it's not actually the net venation. So the venation here is the pinnate venation. So it has the pinnate venation. It has the pinnate venation, very importantly. Now, if you are asked to draw and you draw this diagram, okay, now the margin is entire. So margin is the entire type of margin. Okay, now if you're asked to draw, the first thing you need to do is to write your title. Your title, very important. Then your magnification. How do you get your magnification? You get your meter rule, right? Measure the length of your diagram. Okay, so assuming this length is 6 cm, your physical specimen. Now you're measuring your physical specimen. Then you now measure your drawing on your book. So if your drawing is 3 cm or 6 cm as well, what you do is divide the both value. So this will give you 1. Okay, so your magnification will be written by the side times 1. Very important. So measure your diagram in your note or in your answer booklet and measure your diagram, your physical live specimen, okay, and divide the value. That's how you get your magnification. So that's that. In case you're asked to draw that specimen. Now, other things you need to note is that it has cylindrical stem. It has cylindrical stem, and also um, it has flowers. So it has flowers. It has flowers. It's also it is a dicot plant. I mean, it exhibits epig germination, epigil germination. So these are the things you need to know about specimen C. All right, specimen D. So specimen D is quill feather of domestic fowl. So what are the questions that can emanate from this particular specimen? You can be asked, from which kingdom or from which phylum do we get this particular specimen? So the first thing you should know is that this specimen came from kingdom Animalia. Okay, kingdom Animalia. So, and it came from phylum Chordata and class Aves. So this specimen can be found in birds. Okay, now another thing you need to know about this specimen is how to draw the specimen and how to label it completely. Now, the first thing you need to know is that this part that protrudes out is actually called the quill. The quill. Okay. Then this part that you have this scattered hair, not the one you have the hairs that are arranged and the ones that are not arranged are scattered. So these parts of the quill that is found that the scattered hairs are attached to is called the after shaft. The after shaft. After shaft. Then this part where the arranged ones are attached to is called the shaft. The shaft. And then this part is called the vein. The vein. So these are the basic, this is the basic labeling for this particular specimen. Then don't forget to write your title and your magnification times 2 or times 1, depending on what you have measured. So that is that. So other questions that can come out from this particular specimen, they can ask what's the function or what parts of the organism can you find this specimen. So this specimen is found at the wing region or the tail of the domestic fowl. Okay, it prefers protection. It can also be used for to display courtship or courtship display or courtship display. All right, so that's the function, and this is also where it can be found. So let's write location. So it can be found at the tail region or the wing region. It offers protection and it can also be used for courtship display. So that's that about specimen D.